Hello, guys. Dragon this week. I'm going to go finish chapter 19 for our AMP2 lecture. Now, bear with me. I'm going to get this thing started. We don't like a whole lot here, so we can go into the immune system this week in lecture. Okay, so y'all should be able to see the platelets here. And basically, we just stopped here. The last little section here was, if I can get this thing to work, we were looking at the different types of leukophoresis, which is the production of the white blood cells, right? And you can see this nice two-page picture here showing where they all come from. And we mentioned that platelets are cellular fragments that are going to be involved in what's causing hemophoresis, which is the stopping of blood loss there. And we shall look at that. So here it is. So this megakaryocyte has cellular fragments called platelets, and they're going to stick to the epithelium lining of the blood vessels wherever they're torn, and that causes the platelet plug. And here's the picture I thought I was at there. So here's the left side of the big picture in your book. Figure 1911. There's red blood cells, platelets on the left side, and then the granulocytic neutrophils and eosinophils and basophils next, and then monocytes are agranulocytes, as are the lymphocytes, and they're the B cells and T cells. And we're going to get into this real big in the next lecture next week. Our last chapter for the test, chapter 20, on the immune system. And look at these T lymphocytes are going to do cellular immunity, and these B lymphocytes are going to do what's called humoral immunity, and they produce those antibodies we talked about, you may have heard of before. Okay. All right. And then we're going to go down and look at hemostasis, which is the stopping of blood. I think I may have called it hemophoresis is making of blood. Hemostasis means to stop blood loss. And we got 5 parts here. And actually, I love the Emmer book. There's one part here I don't like. There are examples of these 2 types of coagulation, which is a short answer question that I just changed. So I just updated the chapter 19 short answer questions and put that online. So you may want to go online and look at that one, because I probably will have that one on the test, since I added it after I went over the first half of this in class. But I want to know the difference between the intrinsic and extrinsic pathways, okay? Anyway, so 5 distinct events involved in to form the gelatinous blood clot that plugs broken vessels and limits blood loss, okay? And so the first step's real easy. We have vascular spasms, which we get immediately after the blood vessel is injured. And this is vasoconstriction that increases tissue pressure, both acts to decrease blood flow, decrease blood vessel diameter, but also the blood loss is minimized as both blood pressure and blood flow are reduced by the squeezing of that. So you cut a blood vessel, smooth muscle scraps down to stop it. So these are called vascular spasms, okay? And here you can see it here, blood vessels injured, blood leaks into extracellular fluid, the vessels spasm, and that squeezing down on the smooth muscle and the leaked blood increases tissue pressure, both of which decreases blood flow and blood loss. Okay, so that's the first step. That's pretty cool. Second step, this platelet plug. A patch consisting mostly of platelets adhere only to injured sites, and this further reduces blood loss. Okay, following factors involved in plug formation when collagen fibers are exposed and chemicals are released from damaged cells. These chemicals include this von Willebrand factor, VWF, glycoproteins that bind receptors on the surface of platelet plasma membranes and cause them to produce, exposing the collagen fibers to this chemical together makes the platelets sticky. They adhere to one another into surrounding blood vessels. The binding of this VWF and collagen to platelets triggered a series of events within platelets known collectively as the platelet activation. This includes the release of granules containing ATP, ADP, serotonin, which is going to cause contraction of muscle, calcium, and clotting factors, among others. So that platelet plug is going to clog in that. I always think of this as like the toilet paper when you cut yourself shaving, you put a little bit on it, and it stops the bleeding, okay? The exposed collagen plus this VWF, the granular contents attract and activate nearby platelets, causing them to clump together, not dump. Been a long day. What we call aggregates. So they stick together. This platelet aggregate forms the platelet plug and seals the injured vessel temporarily. Here it is. Platelets coming through. Here's the von Willebrand molecules bind to it, make them sticky, and they stick, and bang, you get a clot there. Now remember, a moving blood clot is an embolism, right? So this is one that's localized. And that stops it. Now, the next step is the one that gets crazy, is this coagulation step. Coagulation is a process that forms molecular glue-like material that binds platelets, endothelium cells, and other formed elements all together. Now, and this includes the plasma proteins that are in the blood itself made by the liver that's going to lead to the total blood turning into gel. If you ever killed a deer and threw it on a four-wheeler and went home and cleaned it and processed it and came back and your four-wheeler has this stuff that you have to get a brush to knock it off. That's coagulated blood. Now, here's the deal. There are two ways this can happen. And I'm actually going to, I think, run off a handout from another book. It's only one of the few times I do all year, but I did pull a handout from another book that shows it as a linear cascade, kind of like a pathway system, instead of her attempt here, Emmerman's attempt at these pictures that don't work real good for me. But there are two of them. There's the intrinsic mechanism and the extrinsic mechanism. Now, my first thought when I heard these as a student was, this must be where they occur at. One's in the body and one's outside the body. No. The two names referred to, and they actually got a second name now to help you understand it a little bit better. The intrinsic mechanism is called contact activation pathway, and what that means is that everything in the blood needed coagulation exists, but it's only activated when it touches a substance other than simple squamous epithelium that lines the blood vessels and the chambers of the heart. So when blood hits the bathtub in your house when you're cutting yourself shaving, intrinsic triggers coagulation when it touches the foreign surface of the bathtub. Extrinsic is tissue factor pathway, is when you cut yourself and the damaged tissues release chemicals that trigger coagulation. So that is this tissue factor produced by damaged tissues, not in the epithelium, but inside the depths of the tissue of the arm or the foot where you pull a nail out. They release chemicals that trigger it. So the source is outside the blood and that triggers coagulation. And intrinsic, the chemicals are all in the blood that is needed for coagulation. However, they both converge at a common pathway that leads to the production of fibrin from the real famous plasma protein made by the liver called a fibrinogen. Now, the best way I can think of this is think of fibrinogen as a soluble form that's totally soluble, not sticky, and then like a duct tape on a spool. But then if you put it into hundreds of strips, it sticks everything. So fibrin is a sticky form that causes the coagulation of the blood. Okay, so fibrin are thread-like proteins that convert soft liquid platelet plugs into a more substantial solid mass. The fibrin is found circulating in plasma and in platelets in an inactive form called fibrinogen. Fibrinogen is converted to fibrin by a series of reactions called this coagulation cascade that occurs at the surface of platelets or damaged endothelium cells. Cascade depends on blood clotting factors, mostly enzymes made by the liver, circulating the blood in an active form. And there's a whole list of them in Table 19.2. We'll see in a minute. Clotting factors are labeled with Roman numerals in order that they were discovered. 2, 7, 9, and 10 all depend on vitamin K for their synthesis. In the intrinsic pathway, named because all involved clotting factors are found in the blood, the pathway activation occurs in following sequence of events. Once inactivated protein clotting factor 12 comes in contact with exposed collagen fibers, okay, or an unknown substance. And I'm not going to look, but the exposed collagen factor activates factor 12 to factor 12A. Factor 11 is then, and 9 become activated. 11A, 12A is enzyme that activates 11A, and you get a chain reaction cascade that leads down to, look at that, it goes on factor 9, factor 8, all this, needing calcium also is going to trigger the coagulation. Here is her attempt to show this. I don't quite get it, but here's the factor coming in, bam, 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 bam. I'm going to give a handout that has it linearly with all the factors leading to this coagulation cascade. And the extrinsic factor, so named because it is initiated by factors outside the blood, occurs simultaneously with intrinsic pathway in the following sequence of events. Damaged endothelium cells display protein called tissue factors. They activate factors 12 to factor 12A. Factor 12A, tissue factor, and calcium ions from enzyme complex activate factors 10 and 10A. So you go down there and you get the activated 10A that triggers the coagulation cascade. Not a very good, some of the worst pictures in the book. I'll give you a handout that shows that. Just know the difference between the two. And then the common pathway, look at this, factor 10A and 5A, and calcium form prothrombin activator, which converts prothrombin into thrombin. thrombin turns fibrinogen into fibrin, which glues the platelet plug together. And there's a nice picture there of a platelet, of a coagulated blood with all the fibrin there, threads holding the red blood cells and white blood cells all together. And that is coagulated blood. Okay. And then it talks a little bit more about that. Okay. And then clot retraction occurs when actin myosin fibers in the platelets contract, pulling the edges of the wounded vessels closer together. Serum fluid consisting of plasma without clotting proteins forced out of a clot during clot retraction, and you get that serum that oozes out of the wound sometimes. Okay. So see here, the blood vessels are pulling in and pulling the wounded edges together. And then the final step is this thromboplastin process that begins, which is going to dissolve the blood clot, because we don't want a moving blood clot will kill you. So this is the process to break down this fibrin glue. Endothermal cells release tissue plasminogen activator (TPA), and it initiates thrombolysis. Urokinase is a similar enzyme produced from the kidney, found in plasma and X-ray fluid, that initiates this thrombolysis or breakdown of the blood clot, the thrombus. TPA activator TPA activates plasminogen, and inactivated plasminogen is found in the plasma. It binds to fibrin molecules as they're incorporated in the blood clot. TPA converts bound inactive plasminogen to activated form of this plasmid that then dissolves the clot. TPA is also one of the most famous drugs of the last 20 years. This is your body's own clot dissolver, and this is given to people who are having heart attacks and strokes. And it opens up the coronary arteries and opens up the blood flow to the brain, and it's a $1 billion biotech drug, TPA. Here it is coming in, binding to the clot and dissolving the clot. Bingo, enough said. Keeping it all together, talks about the two mechanisms here. They both trigger the fibrinogen to be converted to fibrin, so you get a clot, and then we have to break it down by thrombolysis. I think not too bad. Here are all the factors in the order of how they were discovered, but not in the factor and not in the order they're used. All different types of hemophiliac, the inability to clot your blood, is due to birth defects where somebody's missing one of these 13 factors. Okay. Regulation of clots. Some people carry nitric oxide pills that cause vasodilation to open blood flow up. Others have other chemicals here. Heparin is pretty famous that prevents clotting. And here are some of these chemicals mentioned here in the table that can be used: thrombolytic agents and regulators of clot formation. I already talked about bleeding disorders. Hemophiliac is the most common one. A is shortage of factor 8. Hemophiliac B is a shortage of factor 9. So people who have hemophiliac have to have infusions of blood plasma from people, and I think I read somewhere where it's like 100 people to give one treatment. And this is why we had so many of the hemophiliacs get AIDS back in the 80s before we were able to test blood for HIV. So kind of a sad story there. We accidentally had many of them get that and die of AIDS due to giving them bad hemophiliac treatment drug products that contain many people's blood factors, these blood factors. Other ones here: clotting disorders, hypercoagulative conditions. Look at that. Inappropriate clots. Thrombus is a obstructed blood flow, as a clot in the blood vessels, and then if it breaks off, it's an embolism is a moving blood clot. One of the big ones y'all heard about is that deep vein thrombosis. Sit in a car, sit in a plane, sit in a tank for 5, 10, 12 hours, stand up, and you have blood clots forming your knees and other places that bust loose, and you get a pulmonary embolism, go to the lungs and kill you. So these thrombosis in the leg lodge in small blood vessels in the lungs and kill the person. Heparin is an anticoagulant. Morphine's another one. These are some of the things that's they're used in the hospital to get you more, to make you quit bleeding. I mean, they're anticoagulants that's going to prevent your blood from coagulating. And thereby, we give somebody having a heart attack this, so they won't get more blood clots. Aspirin's another one a lot of people take, right? And there it mentions TPA being used as the de facto drug for strokes and heart attacks. Okay, our last part of the chapter is blood typing and matching, and this is kind of tricky because it gets into chapter 20, which we'll hit really hard on Monday, and I'll probably end up shooting Monday and Wednesday, and then shooting a video Thursday and Friday to finish up the chapter on immune system. So I'm going to get a little bit into immunity in this last part of this lecture. But blood typing and matching involves the immune system, okay? Because every cell in your body has markers on them called antigens that your body is going to, your immune system is going to recognize as not being you. And so people who have different types of blood have different markers, and it is possible to share blood with each other because type O, for example, has no markers, so they're the universal donors. And people who have AB have A and B markers on their blood, on their cells, so they can take A blood, B blood, or O blood. So but those of us, like I have O, if you give me A or B or AB, I will make antibodies to attack the A, B, or AB. So those who have no markers or don't have the marker type, like A people make antibodies to B, B people make antibodies to A, and my O blood, I make antibodies to everybody, okay? All right, if that's not confusing, I don't know what is. So we'll go a little bit farther here. So blood transfusion is when blood from a donor is given to recipients. Discovery of separate markers, also called antigens, found on most biological molecules in all cells, including red blood cells, erythrocytes, led to the development of a safer transfusion practice. Immune system recognize foreign antigens and respond to by trying to remove them or kill them or destroy them. This response is the key to these antibodies being made. Now, there are 30 different antigens that we can look at for blood typing, but we're only going to talk about 2 to wrap this chapter up. The 2 blood typing groups, now remember there's over 30 that have been identified, but the 2 that we talk about that have the biggest problem in medicine are the ABO blood typing and RH blood group. Now, so y'all heard about having blood type A, B, or AB, or O. And then the plus minus is whether you have the RH marker or not. Negative means you don't have it, positive makes you do. I believe 65% of people are RH positive, so you have the marker. And we're going to end talking about when that's a problem. And I think I had a short answer question about that one. So take a look. People who have A markers have A markers, have antigen to A. B people have B markers. AB people have both, and O people have none. O type O blood have no markers. Now, in doing this, remember then people who have the A blood here will make antibodies to B. B people will make antibodies to A. So if you give them A, they will coagulate it. AB people make no antibodies to their universal donors. I mean, they recipients. They can take blood from anybody, right? Because they have all the antigens. And O can only receive blood from O, but they are a universal donor. Okay? So we talk about this when we look at the RH factor gets its name from the rhesus monkeys, and they have this antigen D, and so we express that as RH positive if you have it, RH negative if you don't. The most common type of blood is O positive. The rarest is AB negative. And there is some variation in numbers that we see in some ethnic groups. And we can talk about that some other time. The immune cells make actually it's the B plasma cells that make antibodies to attack those antigens. Okay, so as you can see here, the A person here are antibodies to A, and if that happens, if they bump into that, so antibodies to A, that's so a person with B blood here may be given A blood, and here their body attacks it, and you get the coagulation. Okay, and so anti-A agglutinate A antigens, anti-B agglutinate B antigens, and the anti-RH, anti-D antibodies bind to the RH. Here's what we used to do in class. So we would take a petri dish and prick our finger and put 5 drops, put blood in here, and if you get this one right here, you put anti-A in here, antibodies to A, and if they're A antigens, then they coagulate. So that person's A. This one, they didn't coagulate B, so they don't have B. Here, A is negative, B is positive. Here's AB, and then here is O. Nothing happens with O because they don't have A or B markers. So that's a pretty cool little grid there showing that. Okay, pretty cool. All right, and that can lead to coagulation and death of the person, or make them very sick. Okay, blood matches is when you have somebody has the same marker type, or in worst case scenario, you can give somebody O. Okay, and here's showing some diffusion, a blood transfusion. Sue is type B negative. She is her immune system cannot make antibody B because they would agglutinate her own. Sue's erythrocytes like A and H, so she is safely producing the anti-H and anti-RH. But you know, she's whatever. That's kind of a bit weird. Now, probably I'm not even sure if I've ever read that slide before. So anti-B antibodies would agglutinate Sue's erythrocytes. Anti-A and anti-RH do not agglutinate her blood because she has that B negative. And there's tables in here. Here's some tables showing you some of this, and this shows how it agglutinates, right? Donor type AB, recipient O. The O person then has antibodies to AB, and it agglutinates bad. Donor type O, recipient AB, no problem. Okay, so just a couple of those shown to that. O, universal donor, AB, universal recipient. And here's all the combinations and the percentage you can see here in US population with O, the O positive being the most common, et cetera, et cetera. Okay. Moving on to RH factor. RH factor is a problem when, and this is kind of wild. This only occurs when a mother is RH negative and she gives birth to an RH positive baby, which means her husband had to be RH positive. Then, when that baby crosses out of her placenta into the world, some of the baby's blood gets in the mother, and then she makes antibodies to RH. So it's not a problem until she has her second RH positive baby. And when that happens, the RH positive blood crosses into the placenta and causes erythroblastosis fetalis that can kill the baby. So the first pregnancy is not at risk at all, but any other RH positive babies would be at risk. Okay, we have a shot we can give these people developed in the 60s, I believe, called Rogan shot that will make them not make this anymore. So it prevents them from making the RH antibodies. So it's not a big deal anymore because we have this shot. So any O negative woman that gets pregnant, they're going to give her the shot so she don't make antibodies. I had a young girl in my class a couple years ago who had a baby, got a husband, baby's fine, second baby died of this at 8 months in the womb. Kind of find out her husband was RH positive, she was RH negative, she had gotten the Rogan shot and her body didn't recognize it. And then she brought me back, showed me her antibodies, a blood workup showing that she was full of RH antibodies. She was on birth control pill out there. They didn't want to have another kid. And then that semester, she got pregnant again. And that baby was killed within 2 months. So she lost the baby in 2 months. Because every time she was getting pregnant, she made more and more antibodies. And so I even told her, you probably shouldn't have any more kids, adopt or something, or have an RH positive kid, a negative kid, right? So if an RH negative sperm would not be attacked by her body. Pretty fascinating one there. We'll talk more about that in class. All right, guys, I got chapter 19, kind of ran through there pretty quickly. We're going to dive into the immune system. I know Monday I ain't seen y'all in 2 or 3 weeks. Watch these videos. Y'all got to crank it up. And we'll do 20 this week and have our midterm the next week. Remember the midterm for this spring 2021 is going to be short answer questions in person and a multiple choice test question on the night midterm. We're going to have 2 midterms as we already talked about. I'll see y'all later. Y'all have a great day, and I'll see y'all around.